This is your cell and molecular biology course. I'm your course instructor, Inya Central. In the previous video, we explored the discovery of cells and how they were first observed. In this video, we will go deeper into the key milestones in cell biology. From early breakthrough like the invention of microscope to modern advancements such as CRISPR gene editing. Let's begin. The story begins in the 1590s when Zacharias Janssen is credited with creating the first compound microscope. This invention became an essential tool for cell biology, enabling scientists to observe things previously invisible to naked eyes. In 1655, Robert Hooke coined the term cells after observing cork under his microscope. Hooke's description of these tiny box-like structures was the first recorded observation of cells. This marked the birth of cell biology as a distinct field. In 1674, Antony von Leeuwenhoek took cell observations even further by discovering red blood cells and tiny organisms. He called them animalcules. These protozoa and bacteria were the first living single-cell organisms ever observed. Fast forward to 1831, when Robert Brown described the cell nucleus. This was a critical discovery as it showed that cells contain internal structures with specific functions. This further deepened our understanding of cellular biology. The cell theory. Sheldon and Swan proposed that all plants and animals are composed of cells. This idea, known as the cell theory, laid the foundation for all the modern biology. Virchow's cell division. Rudolf Virchow in 1855 further expanded the cell theory by stating that all cells arise from three existing cells. This idea was crucial in understanding cell growth, reproduction, and development. 1857 to 1883 saw major discoveries. In 1857, Kolliker described mitochondria. As we all know, mitochondria is the powerhouse of the cell, and its discovery was a major breakthrough in cell biology. In 1879, Fleming described chromosome behavior during mitosis. This is an essential step in understanding heredity and genetic information transfer. In 1883, August Weismann proposed the germ plasm theory. This explained how hereditary information is passed through germ cells, which are haploid. This concept was pivotal in understanding genetics and inheritance. In 1898, Camille Golgi discovered the Golgi apparatus, an essential organelle involved in cellular transport and secretion. He named Golgi apparatus as Apparato Reticolare Interno. Later, Joseph Nussbaum in 1913 coined the term Golgi apparatus. He included Golgi as a contribute to its discoverer. Mid-20th century advances in microscopy. In 1931, the transmission electron microscope was invented by Ernst Ruska and Max Knoll. This revolution cell biology by allowing scientists to see cells at much higher resolutions than ever before. In 1941, scanning electron microscope was developed by Ernst Ruska. This provided detailed information about the surface of cells. This also allowed researchers to examine the outer structure of cells in remarkable details. 10 micrographic images of grass trim 1930, which was taken in 1933, is shown in the left. Early centrifugation methods of 1950s. Myron Brake's development of rate zonal centrifugation advanced our ability to separate cell components based on size and shape. This method was refined in the 1950s by Matthew Melson and Franklin Stahl, who introduced cesium chloride density gradient centrifugation, which played a crucial role in studying DNA. Here is a cesium chloride density gradient centrifugation figure shown at the left. Discovery of DNA replication in 1953. One of the most important discoveries in cell biology or genetics came in 1953 when James Watson and Francis Crick described the structure of DNA and its mechanism for replication. The Michelson-Stahl experiment later confirmed that DNA replication followed a semi-conservative model. 1981 to 2005 saw key milestones in stem cell and CRISPR research. 1981, Martin Evans discovered embryonic stem cells in mice, setting the stage for regenerative medicine. In 1993, CRISPR was discovered. Francisco Mojica first characterized CRISPR loci and coined the term CRISPR in 2002. In 1997, Dolly the sheep became the first cloned mammal, demonstrating the potential for cloning in biological research and medicine. Ian Wilmette and colleagues at the Roslin Institute cloned Dolly, the first artificial animal clone. In 1998, James Thomson and John Gerhardt isolated and grew human embryonic stem cells in the laboratory. In 2005, Alexander Bolotin discovered Cas9 in Streptococcus thermophilus and identified the protospacer adjacent motif (PAM). 2006 to 2010 saw breakthroughs in CRISPR and stem cells. In 2006, Shinya Yamanaka created induced pluripotent stem cells from adult cells. This bypassed the need for embryos. In 2007, Martin Evans shared the Nobel Prize for the work on stem cells and genetics. In 2008, John van der Ost discovered that spatial sequences are transcribed into CRISPR RNAs to guide Cas proteins to target DNA. In 2010, the first clinical trial using human embryonic stem cells for spinal injury treatment was performed. 2011 to 2014 saw advances in CRISPR and stem cell research. In 2011, Sixney's team cloned the CRISPR-Cas locus from Streptococcus thermophilus, a bacterium resistant to bacteriophages, and expressed it in E. coli, which does not contain a type II system. As a result, the CRISPR-Cas9 locus provided plasmid resistance in E. coli. This confirmed that CRISPR systems are self-contained units. In 2012, human embryonic stem cells show promise in treating blindness. In the same year, Shinya Yamanaka was awarded the Nobel Prize for developing induced pluripotent stem cells. He shared it with John Gurdon. In 2013, therapeutic cloning was introduced. Shokrat Mitalipov produced human embryonic stem cells using therapeutic cloning. In 2014, therapeutic cloning was performed with adult cells. Dieter Egli and Yang Gi Chung created human embryonic stem cells from adult cells using therapeutic cloning. In the same year, CRISPR-Cas9 genome editing was developed. Emmanuel and Jennifer demonstrated that CRISPR-Cas9 can be reprogrammed to target specific DNA sequences for genome editing. Now let's move on to the discoveries from 2015 to 2020, which led to the CRISPR revolution. In 2015, CRISPR-Cas9 was used for genome editing. Feng Zhang successfully adapted CRISPR-Cas9 for genome editing in eukaryotic cells. This demonstrating targeted genome cleavage in mouse and human cells. In 2016, CRISPR clinical trials began. The first human clinical trials using CRISPR-Cas9 gene editing began for genetic disorders. In 2017, CRISPR was used to modify human embryos. Researchers in China modified human embryos to correct a gene defect, a major milestone in gene therapy. In 2020, Emmanuel and Jennifer were awarded the Nobel Prize in Chemistry for their groundbreaking work on the development of the CRISPR-Cas9 gene editing technology. Now let's move on to the recent advances in CRISPR technology. In 2022, small clinical trial showed CRISPR-Cas9 as a promising tool for editing immune cells. This enhanced their capacity to destroy cancer cells. In the same year, the new CRISPR gene editing tools in phages were discovered. In 2023, first CRISPR-Cas9 gene editing therapy was approved for blood disorders. In 2024, CRISPR gene therapy showed a promise as a treatment for inherited vision loss in a clinical trial. From the invention of the microscope to CRISPR gene editing, Cell biology has evolved through centuries of scientific discoveries, expanding our understanding of life at the cellular level. These milestones have not only deepened our knowledge of how cells function, but have also paved the way for groundbreaking advances in medicine and biotechnology. Thank you for watching. I hope you have enjoyed exploring the history and advances in cell biology. Stay tuned for more exciting content. See you in the next video.